Hey, bio students, I'm going to go over a lab that my purple group's doing today and my white group is doing on Monday. And whether you're purple, white, whatever, I still expect you to fill out this worksheet that it's basically a questionnaire. And so if you're 100% remote, you'll just do it based on this video. So here we go. Let's get into this. This is it's actually from Utah, the University of Utah. And it's really good, actually. I've kind of stole some ideas from them. Looking through some of their sites are pretty good. Their packet, I actually printed off this right here that I give students in class. Actually, now I'll probably just upload the digital version since we have COVID and everything. But I got it from them because it's really short, easy, to the point with doing DNA extraction. It shows you that it seems like it would be really, really complicated. And it can be, but it's pretty simple. They break it down pretty well. I will be using split peas, as you see right here, the green peas. I find that works really well because the color. It's a good contrast with the whiter looking DNA that you're going to see eventually, hopefully. So I like that, but they give some other examples of things that you could use: spinach, chicken liver, strawberries, broccoli. I've seen one where the people use a swab of it inside their mouth and try to do that. It works. It's not great, but it will work. Like those DNA test kits, the 23andMe, which I did that. That is just from spit, actually, from your mouth. So the cells from your cheeks and stuff. Now on here, they're kind of joking around. They're like, don't use your dog or your big toe or anything like that. Now, those examples, they would have DNA inside of them, though. So anything living or once living is going to have DNA inside of it. Or at least, now when I say that, yes, that's the problem with dinosaur bones and stuff like that, because they're so old, over 65 million years or more, that the DNA is pretty much dissolved away. Okay, but pretty much it was once living and it had DNA inside of it. Things that do not have DNA are things that were never living, like a rock, like water, like the air around you. Like those are things that just do not have water, or do not have DNA inside them. Plastics would be another example of that. Okay, so when you're thinking of different examples of things that do not have DNA, it's things that were just never alive ever. Okay. All right. So how to do this? If you follow the directions here on the amounts, I find it works pretty well. What I do to try to, use it for the class is I just, double, triple, quadruple the amounts and just the set proportions. And it seems to work out pretty well each time. I've done this for many, many years. So in a blender, it says, a half a cup of split peas, an eighth of a teaspoon, of table salt. And I'll talk about kind of why we're using each one of these ingredients in a little bit here. And then cold water. Cold water just kind of helps keep everything together. The warmer the water, it has more energy in it. That's what really heat is energy. The molecules are moving more. So it kind of breaks it up a little more. So cold water tends to help coagulate the DNA a little bit better, meaning like it clumps together a little bit better, I find. So you blend it up. Now, another thing, so I guess I'll say it this time, the table salt right here, what that actually does, once again, it along with the cold water helps the DNA to kind of come together so you can actually see it. That just is just going to be added to help. All right, now we get into some soapy peas. So what you're going to do after you blend it up for a while, you're going to strain it through and get really the juice of the split peas or whatever you choose to do that was once living. Then it says a couple tablespoons of liquid detergent. Any liquid soap actually does work. I've used dish soap. I've used the clothes detergent. I mean, anything kind of works. You just swirl it. Don't mix it up. Don't shake it really hard. Just really literally just swirl it in there. And kind of what I actually do is I have it in that picture, like you can see pictured right here. And I just kind of swirl it around a little bit like this, just very carefully. So that is good. Now, if you think about why soap, why would you add soap into it? I'm going to ask you, what have we learned about the outside of a cell and every cell organelle, like the nucleus? It's surrounded by a membrane-bound nucleus, right? And membrane outside that's made of what? And hopefully at some point you can remember it's a fat, a phospholipid bilayer, fat. So in order for that DNA to get out of the nucleus, and this is the cell, it needs to break free of the fatty prison it's in here and then it needs to break out of the fatty prison on the prison walls, I guess, on the outside, the gate of the prison, okay? Or the fence, prison fence, probably. All right. So that'd be a better analogy. So that's why we're using detergent, is so that the DNA can actually break free from those membranes. Then enzymes. There's some sites that will say to use enzyme. There's some sites that say not to. I find it works a little bit better. I have meat tenderizer just like that I will sprinkle in. But that's not at the time. That's once you get the soap mixture and all that. Actually, I'll show you here. Yeah, well, once you get the soap mixture, then yes, I actually like to have it go in the fridge for a little bit to even cool it off even more. Because it says like let sit for about 5 to 10 minutes, as you can see it right there. So anyways, I like to have it sit in the fridge. So getting back to the enzymes, here we go. An enzyme, if you remember, is just a type of protein that helps speed up reaction. Okay, so it actually is a catalyst, is the term, that lowers the activation energy needed to get things going. So like it's kind of like the push to get the car going, kind of. All right. So it's going to lower the energy to get things going. Now, this type of tenderizer is going to break up some proteins. Now, the DNA that's stuck inside the nucleus, like I said, is kind of got a straitjacket, whatever my straitjacket look is, like this, of proteins that wraps it up. So it helps it kind of break free from those and start to kind of escape those protein straitjackets. Okay, that's kind of why you use a meat tenderizer. You don't need much. And when you actually, this is a stage right here where you don't want to put too much in, or you don't want to really stir it up too much, because it'll break up too many things. So you just have to be very careful. I literally stir just a little bit as you sprinkle a little bit of this in there. If you don't have this, pineapple juice can work, or I've seen, like I said, I've seen it work without adding the meat tenderizer. So you don't necessarily need to have this so much, okay? And it does say use pineapple juice if you don't have it right there. Okay. Contact lens solution is another option too. Yep, because it's the same idea. All right, so this is the step right here, using alcohol. Now, alcohol is a hot commodity, meaning it's pretty rare right now with COVID. I don't know, everybody's just buying out alcohol, I guess. But if you do have some around the house, what I would do is once I put the pea mixture with the tenderizer in it, I would add just exactly that much of alcohol. Okay, so the same amount of mixture you have, that's how much alcohol you'd want to put in there. And when you do this, as you see in the picture right here, you want to tip the glass. I don't have a glass on me. Here's my Wendy's cup. Okay, you want to tip the glass and pour it down the side of the cup. You don't want to pour it right into it because it's just going to mix up and you're not going to see the DNA. The DNA is very, very delicate. So you want to be very careful at this stage that you carefully pour down the side. And what you'll eventually find is that the rubbing alcohol will actually sit on top of the pea mixture. Okay, so as soon as you do that stuff, you'll start to see some clumps of stuff like this. This is a great picture. So if you did it really, really well, this is what you're going to see. Some stringy, I have to say snot kind of looking stuff. That all is DNA. It's kind of awesome, creepy, weird, whatever, to check that out. But you can touch it, obviously. It's just protein. When you pull it out of the rubbing alcohol, it's not going to last long in the air. Once it dries, it's poof, it's almost gone like that. So it won't last long, but it's pretty cool to check out. All right. Then it just talks about you just completed the DNA extraction. So it's pretty simple. I mean, that's really all it is. It's blending it up, straining it, adding a little meat tenderizer if you have it, carefully adding the rubbing alcohol down the side, and then you'll start to see the DNA. And what will happen first is look like a bunch of clouds on top of the green mixture if you're using peas. And then that stuff will start going up to the top. All right, so it'll rise up because it's less dense. So it's pretty neat. So going over some questions here. I believe I gave you a couple of answers already. I went over how the detergent actually works in the procedure. There's a question that says, what ingredient do you think has the most effect on the amount of DNA you extract? And just explain your answer. So that you can't get that wrong unless you don't explain why you picked something. So that would be either the rubbing alcohol, the soap, the meat tenderizer, the salt. Those are the things that it's asking. And then describe an experiment to test your hypothesis, your idea, number 4. So whatever you did say you think is the most important thing, I want you to just briefly write up a little thing of how you would test, knowing that is the most important ingredient in doing DNA extraction. Okay? All right. I hope that helps. I hope you get a chance to actually do this because it's pretty cool. So anyways, have a great weekend. We'll see you. Bye-bye.